16%. But here's where it gets really interesting. The pathologists didn't just see generic malaria. They saw highly specific morphological features that confirmed our worst fears.
The smear showed delicate ring-formed trophozoites, frequently with double chromatin dots, but most critically, they observed multiple rings inside a single red blood cell.
They also noted appliquiforms, which is when the parasite appears to be clinging to the very inner edge of the red blood cell membrane.
Now, a vital differentiator here is the size of the infected red blood cells. In this smear, the infected cells remain completely normal in size.
Wait, why does the size of the cell matter so much for diagnosis?
Because other species of malaria, specifically Plasmodium vivax and Plasmodium ovale, prefer to invade young red blood cells called reticulocytes, and reticulocytes are physically larger.
Oh, I see.
So if the smear shows enlarged oval-shaped cells, you suspect vivax or ovale, but because our infected cells were normal-sized, it pointed heavily toward Plasmodium falciparum.
And the final piece of visual evidence, they found classic crescent-shaped or banana-shaped gametocytes.
Yeah.
That specific morphology is an exclusive dead giveaway for Plasmodium falciparum.
Alongside the microscopy, the lab utilized a rapid diagnostic test, or RDT.
Right, and the RDT quickly detected the HRP2 antigen. Histidine-rich protein 2 is a specific protein synthesized and excreted into the bloodstream, specifically by Plasmodium falciparum.
Finally, a PCR polymerase chain reaction was sent out to provide definitive molecular confirmation of the parasite's DNA.
But wait, let me push back on this process for a second.
Sure.
If PCR is the undisputed gold standard, and it has the absolute highest sensitivity because it detects the actual DNA, why are we bothering with the operator-dependent microscope or the rapid tests at all? Like, why not just draw the blood, run the PCR, and get a perfectly accurate answer?
Because in severe tropical medicine, time is tissue. PCR takes hours to run, and in many facilities, it has to be packaged and sent to an external reference laboratory, which takes days.
Oh, right.
When a patient is hypotensive, acidotic, and in multi-organ failure, treatment simply cannot wait for molecular confirmation. You must act immediately on the clinical picture, the RDT, and the microscopic smear.
Which brings us directly to the pathophysiology. Now that we have unmasked Plasmodium falciparum, we really need to understand the mechanics. Like, why does this specific species of malaria cause such devastating multi-organ failure compared to the others?
It comes down to two primary mechanisms. Mechanism one is unrestricted invasion.
Okay, so the malaria life cycle begins when an infected Anopheles mosquito bites the host, injecting sporozoites into the bloodstream. These travel directly to the liver, multiply silently for a week or two, and then burst out as merozoites into the blood.
And this is where falciparum becomes uniquely lethal. As we mentioned earlier, species like vivax are picky. They only invade